Hey everyone, we are starting. Hope everyone's having a good day. Hello everyone, my name is Darren Michaels. I am the host of this Scott Labs webinar series where we discuss interesting topics and try and answer questions related to the wine, cider, or specialty beverage production. From yeast and nutrients to stabilization and filtration, we dip into the Scott Labs vault of over 80 years of experience to bring you the latest in research and technical applications. I am your host during this journey, Darren Michaels. I am a Scott Labs fermentation outside technical expert, originally from California, now living up in the Pacific Northwest in the beautiful Columbia River Gorge. It's been hot and windy here, so we all have our noses out for any signs of smoke. Hopefully it won't hit us, but you never know. There was a little fire across the river last week, but it's already been put out, so it's very nice. I want to thank you all for attending and thank you all for taking time out of your day to join us. Today, we're very lucky to have Dr. Nichola Hall, our Scott Labs yeast guru and staff scientist, and all of her vast experience to talk about our budding geniuses, maximizing wine quality with yeast selection, part 1. Nichola started the wine industry in 2002 with an undergraduate degree in microbial biotechnology and a PhD in yeast physiology, first at Bronco as director of microbiology, then as Vincere technical consultant, and finally at Scott Labs as our staff scientist. She is also a past president of ASEV, past chair of Unified Programming, and currently a managing committee member of the Unified Symposium. I want to thank her for her time today. I also want to recommend that if you don't have our current 2020 winemaking handbook handy, that you download a version from our website. Some of the talks this week and next week will reference that handbook. It's really an industry standard to go to when you're thinking about pretty much anything fermentation, even ciders, sparkling. So if you don't have it, just Google Scott Labs 2020 handbook and you should be able to find the link. As I said before, wine is not a simple beverage, and with all the things we can do that will decide the final quality, one of the largest factors is our decision on what yeast to use to ferment our fruit. This is another foundational presentation one I hope you return to as a reference on how to select and manage primary fermentation. Ever since my first my first yeast trial years ago at Sutter Home, I've been amazed and excited about the style possibilities any chosen yeast has on the final consumer-ready wine. Had left Scripps Immunology for the wine industry, I didn't have that much wine experience outside the occasional wine dinner. I understood proper experimental design, but didn't know the importance of yeast selection until tasting those long-term trials. And I was blown away by the impact those yeast on wine quality had up to months and years after bottling. Fast forward to today, and I'm totally blown away by the tools available to the modern winemaker, or brewmaster, cider maker for that matter, when it comes to microbial management tools. As the title suggests, you can not only maximize your quality, but direct wine style. Like using this yeast Sauvy to express thiols for that New Zealand style Sauvignon Blanc, or the yeast CVRP to produce polysaccharides and mid-palate in your Napa Cab. But I also know that before I get those things out of my yeast, I need to follow some basic rules before I can see them. And that's why I'm excited to hear from Nichola today. During the talk, I will be posting several quick polls for you to answer. After the talk, we'll have some time for questions and answers. Some quick webinar tips: make sure you can see the audio settings on the lower left and along the bottom, the raise hand, the Q&A buttons. These will be the main channels for communication. As usual, if you have any questions that occur to you during the talk, feel free to post the questions on the Q&A section. We will try and answer the questions after the presentation. If time is starting to really run out, we do a pretty much lightning round where we just pepper Nichola or any of the presenters with a bunch of questions. Hopefully that's actually a pretty fun time. So please stay tuned for that. Your questions will be anonymous when you submit them. I'll also be looking for raised hands if you'd like to ask your question live. And just so you know, we'll be recording this webinar. We'll be making it available soon after today. I've said this before, sometimes it can feel with winemaking, we're herding cats. In today's case, let's see what kind of kittens we can pick from the litter. Without further ado, I'd like to hand over the microphone to Nichola. Nichola, how are you?
Hey, Dan, thank you. Thanks for a very gracious introduction, and thanks for your excitement about the topic. And thank you to all of you who have joined us for making time in your busy schedule. So as Darren mentioned, we do love yeast and we do appreciate what it can do for us. And if we think about it, yeast are of major economic, social, and health significance in human culture. In fact, we can refer to yeast as mankind's oldest domesticated organism. Yeast has been used in the production of alcoholic beverages and to leaven bread for millennia. But it's really the modern era where yeast has been used to its maximum advantage, and that is to produce medicines, pharmaceuticals, nutraceuticals, in order to produce designer flavors. And next time you have a bag of chips, if you read the back of, for example, sour cream and onion chips, they haven't seen sour cream, they haven't seen onions, but they have had those flavors added to them because yeast can and will produce these compounds when the conditions are optimized for them. Some say the crowning glory of yeast, mainly of Saccharomyces cerevisiae, is it is a model organism in biomedical research. Because it's a model organism, the amount of information we have, it is amazing. So there really are budding geniuses, and today we're really going to talk about how they're budding geniuses and how that applies to their role in winemaking. So with that, I am going to share my screen and I will ask Darren to confirm that we are sharing.
You're looking good. Perfect. Oh, I see your display.
Thank you kindly.
Perfect. Thank you. Looks great.
So part one, yeast management and selection. So the first part of our presentation today is going to be a little bit more academic to give you the foundation to understand more of the impact what you do has in the winemaking environment. So we'll talk about some yeast basics. We'll look very briefly at yeast cytology and physiology. So what this cell looks like and understand what it needs to grow and metabolize. Remind ourselves why we use Saccharomyces and how we can understand what this organism can do and how we can select the best strain for the enological environment. And then Darren's favorite, the lightning Q&A round. So with that, sit back, enjoy, and get your questions ready. So what are yeast? They're not the easiest to describe, but they're commonly referred to as eukaryotic unicellular fungi. And we know a lot about them through how we classify them. So we classify yeast traditionally based on their cell morphology, both the macro and the micromorphology. So macromorphology is how the cells look or how the colonies look on an agar plate. So really what's the shape, the color, the form, the elevation, maybe even the margin. The micromorphology is how does it look down the microscope? What is the shape of the individual cells rather than the colonies? So is it round? Is it ovoid? Is it apiculate? So that means lemon shaped. Is it budding? Is it reproducing? If so, how is it doing that? What is its pattern of reproduction? Is it through fission, which is laying down a cellular plate and kind of growing like in a rectangular shape? Or is it budding? And that is how most cells actually will divide from one cell to two, is through a pattern of predetermined budding. And that budding can be multilateral, so it could bud all over the surface of the yeast. It can be bipolar, so it's only going to bud from the poles. It could be unipolar. Okay, so looking at what the cell is doing helps us classify what that organism actually is. We also spend a lot of time looking at the physiology of the organism. So how are they responding to different carbon sources, different nitrogen sources, different vitamins, different minerals? How are they assimilating them? How are they metabolizing them? How are they reproducing in their presence? How are they able to survive based on what we're doing? So using very traditional methodologies of morphology and physiology gives us a good idea of what the cells are. And we still use a binomial naming system based on the genre and the species. And more recently, we have had genetic tools at our disposal. The tools that we have in the genetic area are faster, they're less expensive, and the throughput is much, much quicker. And by using these very traditional as well as more modern classification tools, we can say, well, there's about 80 different genera of yeast. There's about 2,000 different species, and we're saying about, it's because the natural biodiversity is actually unknown. But what we do know is that we'll find yeast in very varied natural habitats. We'll find yeast associated with soil, with water, with plants, with humans, animals, insects, in the built environment. And by that I mean the winery, the brewery, the bakery, the distillery. We can find them in the atmosphere just above the soil surface. And we can also find yeast in certain circumstances in very extreme habitats, like very high sugar solutions, for example, in concentrates. So because we have been working with yeast for millennia and understanding their role in alcoholic beverage production, we've got a good idea of what they are from a basic level. And if we look at them on that micro level, again, this is what an idealized yeast would look like down the microscope. So we'll see that the yeast on the outside, so this would be the environment, this is your cell wall. And the cell wall is polysaccharide. My little arrows disappeared again. Come on. The cell wall is polysaccharide, and it confers the cell shape and gives protection from the external environment. Then we have our cell membrane, and our cell membrane is a protein lipid bilayer, and that is the site of transport. It is a selectively permeable membrane, so only certain things could transfer into the cell and out of the cell, and this is what confers integrity. Once the integrity of that cell membrane is gone, the cell can no longer function and protect all of its inner organelles. And that is the things that are within the membrane. So the first thing that we see is we have the cytoplasm, and that is the site of major cellular activity. We have our vacuole, which is a storage vehicle in the cell. We have our mitochondria, which is the site of electron transport or energy generation in cells that are respiring. We have our Golgi apparatus and our endoplasmic reticulum, which sorts, modifies, and transports molecules throughout the cell to where they're needed. Our nucleus, and this is why we call yeast eukaryotic organisms, is because they have a true nucleus that contains the genetic material. This is one of the main distinctions from bacteria. Again, we have our mitochondria, which can be numerous in the cell. So this is what an idealized yeast looks like. And if you treat your yeast properly, it will continue to look like that throughout your industrial process, throughout your winemaking, your baking, your distilling. If you damage it in any way, it will start to break down, and it will break down pretty quickly. So as I said, we know a lot about yeast because we have been studying yeast for millennia. It was way back in 1680 that yeast were first observed using a very primitive microscope. In 1818, that was when we discovered or postulated, and that would be the royal we, that yeast were actually living organisms. In 1825, we realized that yeast reproduced by budding, and that was a huge breakthrough of understanding these were living organisms and how they were able to grow in their environment. In 1888, this is when the isolation of pure yeast cultures began. And when you isolate pure cultures, you can separate out what could have been happening by mixed cultures and really determine what is my yeast doing in this environment. And a huge milestone in the world of yeast was back in 1996, when the genome sequence of a laboratory strain of Saccharomyces was fully published. And that strain was referred to as S288C. And when that was published, we realized that there was 12 million base pairs within the Saccharomyces cerevisiae genome. That was 6,275 genes, of which 5,800 were functional, spread over those 16 chromosomes. So this was a huge turning point in our understanding of what our organisms can do. And we're going to come back to that point a little bit later. So our yeast are growing in the environment. So how do they actually grow? Well, they follow a very controlled growth cycle in what we say in a batch culture. And there are 4 distinct phases, and within each phase there are, we can break that down to further. But you'll notice that on your timeline here, there is no specific time associated because it will vary depending on conditions. The number of your cells, again, will depend on your conditions. So this schematic is not exact, but it lets us see overall what is happening. So first of all, we have a lag phase, and that's when we have zero growth. And the cells will enter into lag phase when they experience a change in nutritional or physical conditions. So it's not just when you inoculate them that they'll go into this adaptation phase. It can be any point in their growth cycle when those conditions change so drastically, they have to re-assimilate. How long your lag phase will depend on the growth conditions, the population density, so how many cells there are, as well as the population age. So younger cells will acclimate to those conditions much faster than older cells. But just because the yeast is not growing at this point, there is still a huge amount of metabolic activity going on. Once the cells have acclimated to their conditions, they will enter into their logarithmic phase of growth. They're not Ferraris. They don't go from 0 to 60 in milliseconds. They will go into an acceleration phase, and then they will go into their exponential growth phase. And this, how many times the cells grow, how quickly they grow, will all depend on the conditions. For an example, yeast will divide every 2 hours under laboratory conditions, but in a fermentation, that will take much longer. And that is the difference between idealized conditions and non-idealized conditions. The cells will generally go from 1 to 2, again, keep on doubling, but they will not double indefinitely in what we say a batch culture. The amount of doublings they will do is predetermined based on what we call the cell's Hayflick limit. So in, for example, grape based fermentation, your cells will divide about 4 or 5 times. And once they've divided as many times as that single cell can, they will enter into a deceleration phase, and that is when the cells have experienced a depletion of nutrients, or you have the accumulation of growth inhibitors. So what do they need to grow? What is the requirements for this growth, this doubling via multilateral budding when you are Saccharomyces cerevisiae? Well, we class yeast as chemi-organotrophic organisms, which means they need a fixed organic energy source, or they need sugar. Okay, much easier way to say it. They have very specific nutritional requirements. They require both macro and micronutrients. Yeast macronutrients are nitrogen, oxygen, magnesium, and potassium. Those are what we say are required at the millimolar level. They also require micronutrients, and that's things like zinc, manganese, in certain circumstances, cobalt, sulfur, molybdenum, as well as the vitamins, thiamine, calcium pantothenate, folic acid, inositol, nicotinic acid, biotin, and those micronutrients are required in the micromolar form. They also need the physical requirements to be just so they like their temperature to be kind of 90 is ideal in a non-alcoholic environment, much lower when you have other stressors present. They do like things to be on the slightly acidic side. pH 5.5 is ideal for yeast. And hey, water is life, right? So your yeast do require water. And when you give them all of those things and you treat them properly, that lag phase will be minimized and the log phase will be optimized. So once they come out of that log phase, because they've either run out those nutrients or there's been another inhibitor that is putting selective pressure on them to stop growing, they enter into what we call the stationary phase. So you might have no biomass, so the cells are not dividing any longer, but they're not completely shutting down either. What they're doing is they're opting out of the cellular division, which we say they're going into a G0 phase. And what that tells us is that they're really going to slow down their metabolic rate. So they're going to slow down the uptake of sugar, and they're going to slow down the uptake of their other essential nutrients. We've got a very low rate of protein synthesis during the stationary phase in comparison with the logarithmic phase. And really it is during this phase where the survival factors that was produced earlier in the yeast growth cycle are really essential. So as the cells are opting out and they're staying in the stationary phase, we have the buildup of ethanol in our alcoholic fermentations. So we need to have our survival factors, also known as unsaturated fatty acids and sterols, to protect that plasma membrane. The last phase is the death or decline phase, and yeast, they're not like multicellular organisms which are alive and then die. The cells will go into a very highly controlled set of enzyme-mediated reactions that causes cellular autolysis or cell death. And those can be induced by different factors, whether they're physical, chemical, or biological. So thanks to some of the early work that was done by these yeast pioneers, we actually know how they are growing and how they are dividing. So another huge factor in our yeast history lesson is in 1980. So we're kind of going back a little bit, and that was when Müller-Thurgau introduced the concept of inoculating wine with selected pure yeast cultures, and these yeast cultures were actually grown up from slants into a liquid phase. Then in 1965, to me, this is one of the revolutionary dates in modern enology. So yeast might have been making wine for millennia, but this is when we actually become more involved as winemakers. And in 1965, there were 2 active dried wine yeasts that were produced commercially, and that actually was in California. In 1973, we added another 4 strains. Those of you who have spent longer time in the industry than I might recognize the 4 strains. They were Via Dry PM, Via Dry Epernay 2, also known as CEG, Via Dry Champagne, and Via Dry PM. And this is a huge part of the Scott Laboratories history, because when Scott Laboratories purchased Berkeley Yeast Lab, with that came the Fessler collection of yeast. And it was Robert Scott that worked to bring these 4 active dried strains to market in 1973. They were dried by actually a brewery, and then the year after, 1974, 1975, was when we partnered with Lallemand to bring active dried yeast strains to the market. Then in the 1980s, we introduced strains like 71B, EC118, and K1. And from then, there has been a lot of strains introduced into the market. So it's been in the history of yeast and in the history of winemaking, the advent of enological strains in an active dried form is a relatively recent innovation. So we've gone from about 6 strains to more than 300 strains of enologically important yeast in about 43 years. So out of all the yeasts we could have chosen, I said right at the beginning, there's more than 8 genre of yeast. The genre would be Saccharomyces or Candida or Hansenia spora. Why did we specifically choose Saccharomyces? Well, Saccharomyces does very, very well in a limited oxygen environment. There are no yeast strains that are truly anaerobic. They all need a little bit of oxygen, but Saccharomyces does well in low oxygen environments. Saccharomyces does well in the presence of SO2 up to a certain degree. Although their preferred pH is around 5.5, they do well in our enological pHs from 3 to 4. Saccharomyces does well in sugar concentrations, and it is the actual sugar concentration rather than the oxygen concentration which will dictate its metabolism. So in the presence of sugar above 9 grams per liter, Saccharomyces will preferentially ferment, even in the presence of oxygen, rather than respire. And it does well in the presence of alcohol. So it really is a Saccharomyces species. SPP is referring to all of the different species of Saccharomyces. It really does well in our niche. So how do we get new strains into market? What is the process that they do go through? So first of all, it is isolation. We think we have something of enological interest that does well. So we isolate strains from ferments that have grated from the natural environments, and most of the strains are natural isolates. Step 1 is a genetic fingerprinting. As I said, in 1996, the first Saccharomyces cerevisiae genome was produced, and in 2010, that's when we really started to look at the genetic fingerprint of enologically significant yeast. So first thing is we look at their genetics. Is this yeast truly unique? Then we look at its genetic stability. If a yeast is going to divide 5 times on average during an active ferment, we will look at its possibly 10 times reproductions and see, has the genome changed at all? Is this still the same organism on the genetic level? A lot of strains fail at that point. A lot of strains are not genetically stable. Then we look at fermentation kinetics and its sensory contribution. We want to know how it's going to react and what is its tolerances. And it's its tolerances and the interpretation of those tolerances we're really going to focus on throughout the rest of this presentation. We'll look at its sensory impact. Can we dry the strain? Can we get it into an active dried form without affecting its genetic stability, its fermentation kinetics, and its sensory contribution? A lot of strains may fail at that point. Can it be produced on a large enough scale to make it into the industry? We also do a lot of trials at this point to make sure it is doing what it says it is going to do before it's released to the market. So from isolation to market release can be about 7 years. It's not a fast process, or if you're doing your due diligence each step of the way, it should not be a fast process. So our enological yeast, and when I say ours, I'm talking the ones within the Scott Labs portfolio, are very highly characterized so that a winemaker knows what he is getting. And as Darren said, the 2020 Winemaking Handbook can be downloaded either from the from the website homepage, or you can Google it. And our yeast selection charts our yeast selection pages, are page 10 to 40, and the charts are page 11 to 16. And in there you'll find the yeast selection charts for whites and rosés, reds, as well as hybrids and American varietals. So this is what our yeast strain selection chart looks like. I hope everybody has seen this before. So we have the yeast strain names. Then we go through what they are as well as their technical attributes. So let's look at each one in turn. So the first thing we do is we classify them. And we'll have them as S. cerevisiae or S. cerevisiae bayanus. Now earlier, I said we do genus, which would be Saccharomyces, species, which would be Cerevisiae. So what's with the Cerevisiae and the bayanus after Cerevisiae? What does that mean? So back in the day, right, when we were doing our phenotypic identification using morphologies and physiologies, pre-1978, we had 6 different species within the Saccharomyces genus. In the 70s, we had 3, with Cerevisiae, bayanus, uvarum. In the 80s, they were all reclassified into Cerevisiae. In 1996, remember, the yeast genome was published. And in 98, taxonomy was really the advent of the molecular area. And we had Pastorianus, Cerevisiae, and bayanus. But there was no Cerevisiae or Cerevisiae bayanus in there. So in the year 2020, what does that mean? It means that in the past 25 years or so, there's been so much information on fundamental discoveries that furthered our understanding of our organisms, of our Saccharomyces, our non-Sac, our Enococcus, even our noble varietals. And the genome sequences have really helped us look and see how similar these organisms are or how different. And we group them together on their similarities. And we separate them on their differences. So the first thing we have in that chart is Cerevisiae. So that means it's a Saccharomyces is a genus, Cerevisiae is a species, and Cerevisiae is, we can call it a subspecies or a race, that we will use the term interchangeably. And a Cerevisiae can utilize galactose as a carbon source in those assimilations that we do as part of the taxonomic classification. A Cerevisiae varietal is it cannot use galactose. So what about those Trubayanus from back in '98 when we had those? Well, a lot of them have been reclassified, and only a few very true Bayanus strains exist and are available. They're extremely high in their nutrient demand. They have a very low temperature tolerance and relatively low ethanol tolerance. So in today's nomenclature, we talk about Cerevisiae or Cerevisiae Bayanus, or in Europe they'll call it Cerevisiae galactose positive, Cerevisiae galactose negative. So for me, it's not a huge point that I make. What is the point that we want to think about is, what is the role of these organisms in winemaking? Well, it's the rapid, complete, and efficient conversion of sugar to ethanol, CO2, and other flavor active molecules. Okay. So the next part of our chart is we look at alcohol tolerance. How do we calculate that? Well, it's calculated from fermentable sugars. If we actually know our fermentable sugar, which is in grapes is glucose and fructose, expressed in gram per liter, if we divide that number by 16.83, we should get our potential alcohol. Now, if you use Brix, it's very difficult to do so because Brix does not equal glucose fructose. Brix is a great monitoring tool. Brix is total soluble solids, which is a density measurement impacted by pressure and temperature. If Brix were to equal glucose and fructose, we would not see this chart that I have taken from ETS's Harvest Guide last year. If Brix in grams per 100 mils equaled glucose and fructose grams per liter, 1 Brix would equal 10, and that would be at this bottom line here. We have things above the line showing the relationship or the lack thereof relationship. So yeast is not actually producing more alcohol than it used to. They're not more efficient. It's the level of glucose and fructose or the level of ripeness we've been picking at has changed. Therefore, the amount of fermentable sugar has changed. So yeast will take up the sugar. They will put a little bit through the glyceropyruvate pathway, especially in the presence of SO2. But the majority of sugar is converted to alcohol through glycolysis. Okay, so you want to be able to calculate your alcohol range based on your basic sugar so that you can choose a strain that's going to be able to tolerate that environment. Because you'll see in the chart we have things from 14% alcohol all the way to 18%. So relative nitrogen needs, that's the next major point. And again, you'll see that we class our yeast as low, medium, and high. So how we translate that is that depending on how much sugar that yeast has to consume in that environment, they will need different amounts of nitrogen. So a low nitrogen requiring strain, for every gram per liter of sugar it requires, we need 0.75 parts nitrogen. A medium, 0.9, and a high, 1.25. So this is looking at two parameters for calculating nitrogen needs. It is looking at the yeast needs as well as the sugar level. What this is not looking at is the impact of different forms of nitrogen and how you can actually supplement accordingly. And that can be found in our handbook. So again, we looked at this yeast growth cycle, and how do we look at that from a fermentation standpoint? So this little schematic kind of shows you what is happening. So during our lag phase, our red line being our population, we're not growing. As we discussed earlier, our sugar is not being consumed, but our nitrogen is being consumed. And we've got about 1/3 of the nitrogen consumed during the lag phase. Incidentally, during the lag phase, we also have a huge amount of vitamins and minerals consumed. As the yeast is growing in its growth phase, by the end of the growth phase, which corresponds with the population maximum, we've got, easy math on my sake, 50% sugar consumed, but about 100% nitrogen consumed. Okay, how long it takes for that to happen, as discussed earlier, depends on the population, the health of the population, and the conditions. So what nutrients do you need to add when? You need to make sure at the onset of fermentation, which we say is 2 to 3 Brix drop, the cells have enough vitamins and minerals as well as nitrogen to get through the active growth phase. It's at this point you want to give them their sterols and their unsaturated fatty acids as well, so they can survive in the stationary phase. And at a 3rd sugar depletion, they're going to require nitrogen. Generally, they're going to need nitrogen irrespective of how much nitrogen you start off with. What will change is how many cells you have, how hot your fermentation gets, and the time it takes to get to a 1/3 of the way through. So that's what the yeast need and when and why. And putting it into practice, you know now, looking at your sugar and your yeast, how much nitrogen you need, you look at how much you have and you calculate the difference. And that is how you use this chart, which is fully explained on page 44 in the handbook, okay? We're getting our vitamins, minerals, sterols, unsaturated fatty acids in there from our Go Firm Protect Evolution, and our nitrogen as well as natural vitamins and minerals from our Fermade O and supplemented from our Fermade K. So next up on the chart is temperature. I'm sure everybody listening in today, this is not a surprise that temperature has a huge impact on fermentation kinetics and outcome, okay? What we say a good fermentation rate is about 2 to 4 Brix per day. We know if we're controlling things at that phase, we're not asking the yeast to slow down too much or speed up too much. So we're having nice controlled metabolism. What we will say is that temperature and alcohol have a very similar effect on the yeast. So in the presence of ethanol, you'll see that we don't want to be above 90 degrees in general. So you'll look at your temperature range and ask yourself, can I work within these temperature ranges? Can I heat or cool to keep within these ranges? And if not, I may have to choose something that is going to be a little bit higher in its tolerance. Some of your DV10s, your EC1118s will be a little bit higher. 110 is sublethal in the absence of ethanol, and 115 is lethal. So temperature management is of huge importance. And then just a good tip, you never want to ferment at either end of the yeast temperature range. You want to kind of have it a few degrees off either side. You want to measure your temperature under the cap. Temperature can become striated within about 8 hours. So we want to make sure the temperature we're measuring is the actual temperature in our ferment. And we want to avoid abrupt temperature changes. If we measure our temperature of fermentation, it's again a little bit warm, you don't want to rapidly chill it. Likewise, if it's too cold, you don't want to rapidly heat it up. So remember, high temperature quite possibly can give you high levels of cell death, depending on all the other conditions. And if you've got a lot of dead cells, you quite probably will have problems with your ferment. The last thing we discuss with regards to our yeast strain selection is its sensory impacts. And you'll see that we class things as neutral, enhancing varietal characteristics, producing esters, or having an impact on mouthfeel. So let's kind of look at very briefly at a very 37,000 feet view how that works. So you'll have some winemakers that say yeast do not make a difference. They have no sensory impact at all. And to a certain extent, I will agree with them. There are strains that are sensory neutral, which means they are going to go to sugar, to alcohol and CO2 with no sensory deviations using our little yeast cell here. So sugar to alcohol, no sensory deviations, will give us our neutral strain. So your yeast is not having an impact, and that might be what you want to choose. But the majority of enological yeast do have an impact, and that impact can be seen long term. So yeast will enhance our varietal characteristics, and I'll give you some examples on terpenes as well as thiols. So terpenes are found in the skins, and our terpenes are things that are floral, citrus, kind of woody in nature. They're found in grapes, and they're actually found in hops as well. So floral and citrus compounds are terpenic. Thiols are what we call tropical notes or citrus notes. They're organosulfur compounds. The 3-MH is what we say is this citrus tropical, is found in the mesocarp, in the pulps, whereas your 4-MMP is what we call a vegetal thiol, something that's like gooseberries or grass or boxwood. That compound is found in the skin. So you've got to have a little bit of skin contact there to reveal these things or get them out into your juice. So let's first of all look at terpenes. So terpenes, the majority of them are in the non-odoriferous form, which means we can't smell them, and they've got to be released to the odoriferous form so we can smell them. And it depends on if the terpene is bound up to one sugar compound, 2 or multiple, or more than 2. If your terpene is bound to one sugar, it is generally bound through to a glucose, and your yeast, your Saccharomyces cerevisiae, because they all have different genetic makeups, and we fingerprint them to see what the genetic or what the genes are coding for. If we have a monoterpene, so a terpene linked to one sugar, and that sugar is glucose, our cerevisiae will produce beta glucosidase that's active during the fermentation to reveal the thiol. If it is linked to more than one sugar, your Saccharomyces has a little bit harder time. Flavia, the non-sac, being a Mechnicovia pulcherrima, have at it all day. Does a great job producing the enzymes that will break those down. And our little friend bacteria, Enococcus, has the ability to reveal terpenes. So depending on the varietal, is it a terpenic varietal, and the yeast you choose, you can impact the terpenic expression. Yeast is also thought to produce some of them de novo, so on their own. A little bit less is known about this. So the thiols, how do yeast impact thiols? So you've got to first extract the thiols there, but your thiols, like terpenes, are generally in that non-odoriferous form, so we can't smell them. Unlike the terpenes, which are bound to sugar, your thiols are bound to nitrogen-based compounds. It can be bound to a cysteine, or it can be bound to a glutathione, which is a tripeptide containing cysteine. Your cysteinylated compounds are taken up through through the cell wall, across that membrane, that site of transport, through a transport system that we refer to as GAP1. If you have too much ammonia in the system, the cells will preferentially take up the ammonia and will induce something called nitrogen catabolite repression and will not take up these amino acids. So they will be left out in the grape juice unconverted. But your yeast can take them up, and depending on the yeast genetic background, whether they can release these things. So we see the pathways here for the succinylated revelation as well as the glutathione thiol revelation, because they're taken up through a different system. It's thought that to release 4-MMP, or it's known, I should say, to release the 4-MMP, the yeast actually needs to have a specific gene encoding for IRC7. And only about 7% of the enological strains actually have this. So not only do you have to have the thiol present, you have to have the yeast take it up, and the yeast have the correct genetic makeup to reveal it, okay? So we've got to have a lot of things in order for the yeast to express this thing that we call a grassy thiol or a gooseberry, whereas for the 3-MH, the tropical citrus notes, it's a lot easier. And we can see in this graph, this is taken from our 2020 handbook, the impact of the yeast strain QA23 versus Savi on the 3-MH, tropical citrus notes. The esterified form of this compound, so some yeasts are releasers of thiols, some convert the thiol into the ester form, as well as that 4-MMP. And I also popped in there the sensory thresholds in the green to show the impact of two different yeast strains on the final thiolic expression in your wines. Oh, so esters. Okay, I said yeast can produce esters. So those are the fruity floral compounds as well, different from the terpenes. And yeast are thought to produce esters as a means of detoxification. So these are something produced de novo by the yeast, and they could be produced via carbon metabolism or through nitrogen metabolism. And depending on the genes that the yeast is producing, whether it's the ATF1 and 2, the aminotransferases, we can produce acetate esters, or depending on this set of genes, we can produce ethyl esters. Acetate esters were generally at higher levels than the ethyl esters, because the ethyl esters, they don't transfer very well across this lipid membrane. So the yeast strain has a huge impact on ester production based on its genetic background, the temperature of fermentation. This is showing some recent work conducted by INRA as well as Lallemande, looking at the nitrogen level as well as the natural grape sterols on ester production. And this is 7 different yeasts using the box Banham designs. And I don't expect you to look at them, but if you could kind of see the different patterns. And where it's hitting its peak is where this compound specifically is ethyl hexanoate, which is general fruitiness, is produced. So a similar-ish pattern for 71B and CY with regards to nitrogen, but different with lipids. And the others all showing the differences. So your yeast, depending on how you handle it and what it's exposed to, will produce these different things. So kind of almost there, sensory impact on mouthfeel. So I said at the beginning, the yeast cell wall gives protection to the cell. It gives the cell shape, and it was polysaccharide based. Well, in general, it's metaproteins, glucans, and chitins, and these are what we call polysaccharides. Okay, so multi-branched sugar compounds, some of them interspersed with proteins. And different yeasts will have different metaprotein signatures. And the different metaprotein signatures and the different release of those different metaproteins will impact the mouth. So you can see here some work that was done and produced in the Yellow Journal using RC212 and BM4x4, how much polysaccharides is produced over the course of the fermentation, up the end of fermentation and during autolysis. And we can see that BM4x4 has a very early release of its polysaccharides, so it could interact with our tannins, leaving less points of contact in the mouth for our tannins. So they're kind of working alongside our tannins to soften them up, giving us roundness. So over the past 45, 50 minutes, we've gone over how to select your strains for success. We haven't really gone over competitive factor. There's a lot of conflicting information. The yeast will produce mycoproteins. They could be mitochondria or cytoplasm derived. That they're somewhat active in the enological environment. I don't pay that much attention to it, and I haven't really seen a huge impact of it on fermentation outcome. So with that, we've looked at what yeast are in a very broad sense, how they grow, what they need to grow and survive in the enological environment, how this information applies to enology, and how you could use this information in order to tailor your yeast selection. So how can you use the information we present to you through our handbook in order to make sure you are successful? So with that, thank you so much for your time. We greatly appreciate it. And from all of our fermentation and enology team here at Scott Labs and the company at large, we really want everybody to have a really safe and a very successful harvest. And Darren and my email is up there if you do have any follow-up questions. So with that, thank you again. Darren, back to you.
That was great. Thank you so much. Fantastic as usual. Yeah, this is scheduled till noon. We have some follow-up questions submitted by the questions and answer board. So if you have to go, though, I understand. Thank you again for coming today. This was part one. Remember to show up next week for part two. If you haven't registered, tell your friends, or go ahead and register for part two. Same time, same day, next Thursday, the 13th. And I really do want to thank you all for showing up. I know it's a little bit of your time every day, but we really do appreciate it. I'm going to go ahead and start with some of the questions, and then if we run into like a lightning round, we'll do that, and then I'll finish up with some of the poll results. So thanks again. That was awesome, Nichola. Thank you so much. So one of the first questions we have is, have sugar to alcohol conversion rates changed over time? You know, we used to use an average of 0.58 or 0.60. Have you seen anything change, or what have they? What's your feeling on that?
You start off with a certain amount of sugar, and if you know how much sugar you actually have to ferment, your stoichiometric reaction tells us no, that is not true, and I will stick by that. Now, depending on what you're asking the yeast to do, you'll have slight variations, but nothing of significance. Okay. Now, if we think back in the day, right, when we were picking at 23 bricks versus today picking at 27, the sugar profiles may very well differ, right? Under 24 bricks, you might have a little bit more glucose. Over 24, and definitely once you're getting up to that 25, 26, you might have a little bit more fructose, but that does not change that glycolytic flux. Now we do have some strains in our portfolio that we've made change that glycolytic flux, like Ionis, that instead of 1 gram going, it's doing a little bit less.
Yeah.
Saccharomyces in general, no.
So pretty static. We did find too, because the conversion rates, I was going from a large winery, and then when we started to do stuff at Terravant, I went back first year review and I started to see a wide range of conversion rates. But then when I started to isolate which ones were open top punch down tanks and which ones were closed, very cold fermentation, my conversion rates ranged greatly. I mean, we had ethanol removal systems, so the open tops, usually the ranges were around, conversion rates around 0.56, 0.58. But then the cold, very cold fermentations, very tall tanks with closed tops, those conversion rates, quote unquote, were much higher. So we saw a lot of conservation of the ethanol. So a lot of that, I think, is also thrown in there too.
Yeah.
Like, how are you conserving the ethanol, if any? What about Bionis designation? When I started the industry, or 20 years ago, and then I still get questions. Well, is that yeast strain Bionis? Is Bionis like it's a super bug? Is that designation important anymore? What's the current status of Bionis?
There are, I think, about 4 true Bionis strains in the market, and those strains have an alcohol tolerance of about 14%.
Okay.
Because of the advent of genetics, we are now going genus, species, subspecies. Now, there could be a difference between the subspecies other than the galactose metabolism that we have not found out yet. I don't think it is a part of the subspecies designation, but we will see our Cerevisiae Bionis having slightly higher alcohol tolerance, but Bionis is not more tolerant.
Okay.
So if you're looking at all the other parameters, what is the alcohol tolerance of this organism? What are the other technical parameters of this organism? The name, we can argue, doesn't matter.
Okay.
The name should not be the deciding factor.
That's a shift too. Yeah. Cause I'm for years now, it's like, I need a Bionis. So I'm like,
what do you actually need? It's not the Bionis you need something, potentially it's 16 alcohol.
Gotcha.
Right.
So yeah, look for that alcohol tolerance basically. Is there any tips that you have for maximizing or minimizing autolysis, certain strains?
Minimizing autolysis?
It says maximizing or minimizing, changing autolysis.
Okay, so these strains all have a predetermined autolytic rate. The strain with the fastest autolysis rate that I'm aware of is CY3079. As soon as one cell has done its job, it will start to die off and break down and release its nutrients. But most full autolysis takes 8 to 12 months. The enological environment's not conducive to autolysis because of our low temperatures and low pHs. So we can use our beta-glucanase enzymes in order to break down that glucan fraction of the yeast cell wall, helping to expose the internal membrane to ethanol and pH, which will speed up the autolysis, as well as stirring. The more you stir, that mechanical action will help with autolysis. But for use of enzymes, it's about 6 weeks.
Okay, that's not bad. And I know we got a million, well, not a million, but we got enough yeast strains in our portfolio that it'd be hard to answer this question. But I'm just going to throw it at the top of your head. Yes, exactly. This is a terrible question.
Cheers.
At the top of your head, what would you recommend for hot climate Tempranillo and Syrah to maximize mouthfeel? And it says thiols, but like maybe esters. Any off the top of your head, what's your go-to?
So Tempranillo and Syrah?
Yeah.
I don't know if those of our viewers say no. Our very good friend Pepe, Jose Maria Heras, he is our Lalamand, Spain. His girlfriend, he said, I don't know if his wife knows, is RP15 for those varietals for those reasons. I like the Spanish varietals. They do great with our hot climates. They help them open up. They're low nutrient demanding, high relative alcohol, and wide temperature range. So things like VRB T73 Clo.
Okay. What are they? Okay. Okay. And always everyone, look through the book too. I mean, there's, if you've got any serious questions, please don't hesitate to call us or email us. But a lot of your questions can be answered by that handbook.
And remember, Darren, we only spoke about active dried yeast today, right? We've got a whole portfolio of inactivated dried yeast. So if you need to build more mouthfeel, that's what Optimum Red is for.
Yeah, gotcha. Yeah, totally, right. And
one comment. Yeah.
Oh, I was just going to say really quick with the maximizing or minimizing autolysis, don't forget and don't underestimate the power an enzyme like Lauzyme MMX does for it's a glucanase, and it will really affect. And a friend of mine who's done dry Sauvignon, dry Rieslings, which is not a huge market out there but the hardcore really love them. And he'll do 9 months aging Sir Lee's, and he uses Lauzyme MMX glucanase to build it. And in a controlled experiment, it was crazy. The Riesling tasted like it had sugar in it, and it did not. Now, here's a ringer question. I keep this for last, and then we're going to go through some polls. So thank you again, everyone, for showing up. You have to leave. I understand. I really appreciate it. Don't forget, next week is part 2. Now, here's a question, and I don't understand the question, so I'm just going to read you the question because I'm not. Not smart enough to, but I'm gonna. It's quite a question. So if you need telomere damage or cell membrane limitations, what is the Hayflick limit?
Sorry, I missed the start of the question.
Okay, is the Hayflick limit related to quorum sensing? So first of all, what is the Hayflick limit? Does that sound familiar to you?
So the Hayflick limit is the amount of times a cell will divide in a given environment. The enological environment is 4 to 5. And the reason it's 4 to 5, and this will be a long answer for a long question. Our yeast cell has about 5% lipids in that phospholipid bilayer. Each time the cell divides, those lipids will reduce by 50%. Once we get to 0.125% lipids, the cell membrane can no longer confer integrity. So, right, 5%, 2.5%, 1.25. So once you hit that 5%, that 5 doublings,
you're done.
If you make your cell do any more, you'll have problems getting to stationary, and you'll have an early decline phase and a lot of sugar left.
Is that, and the question really is that from quorum sensing or telomere damage or cell membrane limitation? Is it cell membrane limitation?
Yes. Well, it's going to be intracellular damage because the membrane is damaged and it's no longer selectively permeable. So everything's going to flood in, all that pH. The cell can't shun out those hydrogen ions. It can no longer maintain intracellular homeostasis. So it doesn't have anything that I'm aware of to do with quorum sensing. It's more to do with physiology.
Well, I'm glad you answered that question. I was a little flummoxed on that one, but I appreciate it. There's a couple. So guys, there's a couple more. Here's some results from the yeast differences poll. Do you think yeast selection makes a difference in final wine quality and sensory impact? It wasn't even close. It was 98% said yes. Just so you guys know, when I get about 50% responses, I turn off the polling just so I can get the poll out of the way. But 98% said yes, you do think selection is important. And then the third poll, the first poll is about Scott Labs history. I do appreciate a lot of you. Here's the first poll, in case you're interested in how we answered that. Most of you, 40%, knew that Scott Labs was instrumental in bringing dried enological yeast to the worldwide wine industry. 10%, 25% didn't, and then the rest of you just found out. So I appreciate just finding out. Again, thank you for showing up. Last one, yeast selection. Do you tend to select a particular yeast for its technical attributes, alcohol tolerance, temperature range, etc., or its sensory impact? it was almost split. There was a slight increase over sensory impacts, but then there was a fair amount that they pick it over technical attributes, which is a very, very good answer. I'm happy to see that a lot of you understand that. And we can't stress enough, and I think Nichola would be definitely one to back this up, is that we really stress that if you're going to maximize your quality, and that's what's really this is talking about, maximize quality and push your wine style, that you really get down the first technical attributes of whatever yeast strain you pick. Because, and correct me if I'm wrong, Nichola, but I mean, if you don't get the right temperature range, if you can't control it, at least pick a yeast that'll put up with that. If you don't have the ability to modify your nitrogen levels, if it's required, or even testing it, and if you don't know what your alcohol tolerance is, the major big three, you really can't hope to get all of what you want out of the yeast. Is that fair for me to say, Nichola?
Yeah. You want to secure your fermentation. That should be our number one goal. And then drive wine style.
Yeah.
And you could do both simultaneously, as long as your selection is conducive to doing so.
Yes, perfect. All right, well, thank you guys. Well, thank you, Nichola, for your time. That was awesome. Loved it, as usual. I think that's it. Thank you again for your time. I can't stress enough that all of you, thanks enough that if you get a chance, please submit a survey to the survey. There might be a survey that's available for you to fill out. I'm not entirely sure this time we're going to do that, but if it is, please fill it out. Our upcoming topics are based in your comments, so keep them coming. Today's topic and next week's topic was directly related to the requests made in that survey. It was the top 1 and 2 was how to do yeast selection and how to do nutrient management, and that's by far the top 2. That's why these 2 came out. So we do read your comments. We will be creating new webinars based on that. And also keep in mind as well, like I mentioned with Maggie's and I mentioned with Luke's and with Nichola's today, these are foundational webinars. We are going to be doing higher level webinars in the future, more advanced topics. Don't hesitate to reach out as usual. Any other questions or comments? We believe in continual improvement. Nichola mentioned it. My email is darrenm@scottlab.com, darrenm@scottlab.com. We posted the webinars on our website, so you could just Google Scott Labs webinars. It should show up in the search results. Also, again, with our handbook, don't hesitate. 2020 Winemaking Handbook, Scott Labs. Put it in Google. Put it in the other one, the Microsoft one, which I cannot remember off the top of my head. It shows up in that one too. Thank you all so much for coming. All of us at Scott Labs, we want to wish you, as usual, wish you and your family health and happiness. And everyone have a great time. Thank you for all of us that are still hanging in there. Thanks, Nichola.
Thanks, Darren. See you soon. Bye.
Thanks.